Fine. All right, good morning, everyone. I am very happy to be here. Thank you very much for standing up this early in the morning to visit our first breakfast seminar. And I am very proud to be guiding you through the launch of two new products here at MSAID that we're presenting here at ASMS 2024. And I will dive right in with introducing the update to our flagship algorithm, CHIMERYS, which is a search algorithm for proteomics that banks heavily on predicted peptide fragment ion intensities. As you guys know, we develop software for proteomics based on accurate predictions. 3 of those are integrated into Proteome Discoverer at this moment, and today I'm going to focus on updates to our flagship CHIMERYS. Martin will dive a lot deeper into how CHIMERYS works, as we just published our first preprint that goes into the technical details on how the inner workings of CHIMERYS actually play together. For now, I will just make you aware that CHIMERYS utilizes predictions to explain the experimental MS2 spectrum in a linear combination of predicted fragment ion intensities. What that means is that we go through the raw file spectrum by spectrum. For every spectrum, we determine suitable candidates that we predict, and then we mix and match these predicted candidates in a way that it best resembles the experimental MS2 spectrum. We hand everything over to Mockapot for FDR calculation, and thereby can dive a lot deeper into DDA data, also process DIA data and PRM data. The feature set of CHIMERYS has been growing over time. So in 2022, we released the first version of CHIMERYS. It was integrated in Proteome Discoverer 3.0, and it was back in the days was only capable of processing DDA data. However, there we doubled peptide identifications routinely in contrast to other classical search engines. In terms of PTMs, we did not support anything besides oxidation, which is why in the next year we focused on integrating more compatibility with more biologically relevant modifications. So in 2023, we released the unifying approach, which enabled processing of DIA and PRM data with the exact same algorithm that we use for DDA, meaning that we deconvolute DIA spectra in a DDA kind of fashion, giving us all the information on a spectrum by spectrum by spectrum level. We also included phosphorylation as modification and paired CHIMERYS together with PTMRS for the localization. And today, I'm very proud to announce that we are releasing CHIMERYS 4, which is going to be integrated into Proteome Discoverer 3.2. It will still process DDA, DIA, and PRM data, and it does that much faster than the previous version. We also added new modifications, acetylation and ubiquitination, so scientists can dive deeper into signaling pathways they could not process before with CHIMERYS. And to replace PTMRS, we developed our own native PTM localization mechanism, which is also now capable of localizing PTMs in DIA data, which was not possible before that. So one of the bigger changes to CHIMERYS 4 comes from our prediction model in Ferris. So over the last couple of months, we spent a lot of time into developing pipelines that allow us to dive into the wealth of data that is stored on PRIDE. Because 4,000 data sets are added every year on PRIDE, and unfortunately, not a lot of that is reused besides a couple of downloads after the initial publication. So we set out to write automatic pipelines that systematically download training data, harmonizes it, deposits it in data lakes, and then this data can be aggregated into training data sets to train thousands of models using new modifications. Upon popular request, we extended CHIMERYS and in Ferris 4 to support unmodified cysteine residues, because that was bound to carbamidomethylase previously. We also included a modification that comes with the Proteomics kit on cysteines, and we were able to generate models in lysine acetylation ubiquitination without using any synthetic peptide libraries as we have used before. If you look at the distributions of these, you can see that the spectral similarity when compared to a holdout data set matches the quality of the predictions of unmodified peptides. And we're very happy about that, because that now enables us to dive deeper into acetylation ubiquitinome data. When you look here at a single-file DDA processing analysis, you can see that we can easily double the amount of spectra that are identified for acetylation and ubiquitination and increase the number of unique modified peptides between 70 and 50%. And this is really cool because all of these low stoichiometry PTMs that are there, and people spend a lot of money on buying antibodies to enrich them prior to any of the mass spec analysis, and now software basically enables to get a much higher yield on the reagents you spend, on the LCMS time you spend. However, PTMs are only fun if you functionalize the position. It can only be functionalized if you know the exact position of these. And this is why we came up with an all-new PTM localization mechanism, which basically uses predicted fragment ion intensities to pinpoint where that modification is. In a nutshell, and I know this is a bit of a complicated figure, we predict all isomers for this modified sequence, and we compare these to the experimental spectrum, and then devise a score that represents a probability out of the spectral similarity and the differences in spectral similarity between the isomers. When you apply this data to ground truth synthetic data, you can see in the DDA case here that CHIMERYS-4 outperforms CHIMERYS-2 with PTMRS and max quant in these precision recall curves. So the precision recall curves describe the separation power of a score, and what you're want to see is you want to see a large area under the curve here. So you can see that the dark blue line here for CHIMERYS-4 clearly outperforms the old algorithm together with PTMRS. On a DIA data set, we can see that PTMRS was unfortunately not written to be able to pinpoint localizations in very complex data, which is why the line for PTMRS and CHIMERYS-4 directly drops to 0 here. However, the new algorithm outperforms also the localization algorithm of Diane, and that should enable people to dive deeper into PTM DIA if they desire to do so. One important point here is that with CHIMERYS, even for DIA, PTM DIA analysis, you still have information on the spectrum level. So if you want to make sure that whatever the search engine told you is going to be in that sample, you can still make sure yourself by going back to the roots and interpreting a spectra manually. Speaking about DIA analysis, we heard the community and worked a lot on improving the workflow architecture to decrease runtimes of the CHIMERYS service. What you can see here are 3 different types of astral files. It's 180, 160 samples per day, which corresponds to 8, 15, and 24-minute gradients. And you can see that on a single file processing, you reduce the amount of processing time that you require by a factor 2. If you then go into multi-file processing, so you have aggregated a larger study, you can see in this chart here on the right-hand side that processing 10 times 24-minute files, so 250 minutes of gradient time, can now be processed way below acquisition time with CHIMERYS-4. And now, at the very least, I want to talk about an important feature of CHIMERYS-4 that's very dear to our heart. As I said, CHIMERYS-4 is going to be integrated into Proteome Discoverer 3.2. But what makes me insanely proud and is a real milestone for us as a company here is that we can announce that we'll make CHIMERYS-4 also available as part of our all-new software as a service platform without the need for Proteome Discoverer that we're launching today. So let me take you into a bit of a journey how we think about this and how we came about this platform. In the last years, instruments and lab workflow have tremendously improved in terms of sensitivity, automation, usability. You need less experts in the field to generate the data. You can have more users in the field. And I think this is really important for us as a field moving forward, that we're basically abolishing the need to have an expert in the lab, an expert in the bioinformatics department, an expert in your infrastructure to generate insights into the data that you generate. However, in terms of what happens with data that rolls off the mass spectrometer, there is still a gap. And let me tell you a story that I experienced myself, how that usually can go, and I'm very certain a lot of people of you have also seen such workflows. So data, in the worst case, rolls off a instrument PC and gets carried towards a processing PC on a USB drive or an external hard drive. That processing PC is shared with all of your colleagues somewhere in your internal infrastructure. And after you processed your files, you'll probably put your results on that USB drive again, and you walk to your analysis PC, which also means that the data is usually stored in the home directory of the user that generated it, and then analyzed on a local laptop. If the scientist is well-versed in Python or scripting languages, he might do a more thorough data analysis. If he's not, he might just look at Excel tables and sort for most abundant proteins. And it's really hard to derive actions out of that in a reproducible manner, also without having the necessary skill sets. So we identified this long-winded path from raw files to insight in action as one of the most critical pieces of missing infrastructure in labs. And we have set out to implement a vision to transform this process. So here I present the integrative platform that covers all aspects from raw to insight. And ladies and gentlemen, we term it the MSAID platform. You can see that we have not slept a lot in the last couple of weeks to get this done, so we didn't have a lot of time to spend on properly naming the thing or coming up with a very fancy name. But since MSAID stands for Aiding Mass Spectrometry, I think MSAID platform still has a nice descriptive ring to it. So what are key features of this platform? Well, it's a web-based platform, which means you can access it from everywhere you have a web browser or internet connection. And if you desire to do so, you can also do that from the couch on your phone if you really want to spend your weekend looking how your searches are going. Since it's a cloud platform, it seamlessly scales with volume, meaning that depending on how much data you want to deposit, how much data you want to process, we can give you a tier that would allow you to do exactly that. Also, it's a cloud service, so we can provide elastic compute on demand, meaning that if you have a larger study you want to process fast, we can do that, just that, without a problem. Also, we control where the data is stored, which means that you can systematically integrate over experiments that you did, over runs that you did. You can retrieve the data, you can query it, and you can build, aggregate experiments on data previously acquired. The platform itself is API controlled and automatable, which means that it can basically interact instead with a web interface also with any existing cloud pipeline that you already have in place. So we believe that this automation process is one of the things that is going to be very relevant for the customers, especially if you already have a lot of data in the cloud and don't want to download it, process it, and move it back up again. And we decided for a subscription-based model to have a low entry barrier for people who want to try it out and want to get their hands dirty with cloud-based processing. So let's look at some of the core features in detail. As I said, you can pump data into the platform directly from your browser, but you can also do that using a CLI client that you can run, for example, on your instrument computer or any other net drive that you have. We'll also plan to support UI-based transfer tools for people who really still want to see that Explorer window if you move data from A to B. Once you uploaded your data, we have free text tags. Jesus, something is moving. Free text tags that allow capturing metadata, descriptions, categorizations, so you can organize your data and retrieve it easily later. That can be done, for example, by just tagging them directly in the web interface, but we also foresee automatic tagging of the raw files with your permission by extracting metadata from the raw files and automatically attaching them to the files, or importing data from the system when you already have set down and annotated your files with metadata that you want to have. So the platform itself and the compute here is fueled by CHIMERYS 4, which means that it can process DDA, DIA, and PRM data in a spectrum-centric fashion. But this platform itself is designed in a modular fashion to cater any search engine, meaning that we just have to write adapters in the front of the cloud and the end of the cloud processing workflow. So if you're a software developer, feel free to approach us, and we'd be very happy to discuss what kind of options we see. And one of the reasons you might want to do is because you can profit off the elastic compute that we can offer in our cloud infrastructure. So if you look at the scalability of 1 to 100 raw files, in this case from an HFX you will see that the processing time between one file and 100 files in the end just increases by 40%, which is mostly due to aggregation steps that we need to perform once all searches are finished, like FDR rollup and quantification. And by the way, while you're processing, you can look at all of these data, including all of the log files, right in the web to see how the progress of your files is. And once these experiments are finished, you can actually do something that is usually not happening when you download your data or store that non-systematically. We can, across experiments and across data that you acquire, combine experiments together because we have all the intermediate data that we require for that still on the platform, which means you can longitudinally acquire data for your study and then in the end do not have to run it one time altogether. But the only thing you do is you roll up intensities, you roll up your FDR, you do your protein grouping, and you're done with that. And that time frame is obviously a lot faster than just going through everything again. And once you're finished with your experiment, we know that a lot of people just want to have a quick look into the data. Maybe you just want to check if your numbers are okay. Maybe you just want to check on whether the QC that you usually perform is within specs. Or you ask the question, is my favorite protein is there? In this case, for example, EGFR, because that's our favorite protein when it comes to biology. Well, you can see that, and you can see all the evidence for EGFR here in this table. Obviously, you're also able to download all of these files, but that's a little more boring. And because we know that looking at tabular data in proteomics is not very pretty. Actually, proteomics data is very ugly. In contrast to people who do like imaging studies, who always have these great covers for the articles they're publishing, we usually have tabular outputs that we sort by some kind of metric. And this is why we implemented a visualization tool, a collection of plots that you can do right in your browser that are interactive, downloadable, and customizable to check certain specs and metrics in your results. These plots will grow over time, also by popular request. We don't have that many yet, but that will happen in the very near future. And with that, I want to stop talking about a website on a PowerPoint presentation because it's really hard to transport how that feels and how that all interacts. And what I want to do is I want to invite you all to try it yourselves. The platform is hosted at platform.msaid.io, and we invite everyone to sign up. We will gradually admit interested parties and testers to the platform. Obviously, testing the platform is covered by us, and we would be very happy if you give us feedback and thoughts on features that you're missing or features that you are very happy about. You can also already request quotes for the platform. We will be very happy to connect our sales team with you to discuss these. If you want to sign up yourself, you can also come by our booth, 200. We're going to be doing live demos of the platform throughout the conference. It's just right in the front of the entrance of the poster hall. If you're interested in a little bit more about the technical aspects of the platform, feel free to come to my poster on Wednesday. And as I said, try it out and let us know what you think. And with that, I want to thank everyone for attending our product presentation before we go into the scientific parts of this workshop. Are there any questions?